Hi guys, this video is about the questions I have prepared, whatever we have learned so far about glycolysis. So let's start with the first question. Which of the following is true regarding the hormonal regulation of glucose metabolism? So as you have remembered, like what I told you in glycolysis part 2 video, that how fructose, that glucose is transformed into fructose 6 phosphate. Fructose 6 phosphate is fructose 2 6 bisphosphate. Okay, and how insulin was responsible for the fructose phosphofructokinase 2 activation, and this whole fructose 2 6 bisphosphate was responsible for the phosphofructokinase 1 activation. Okay, and we also learned that fructose 2 6 bisphosphate, in the presence of glucogen, it transformed back to the fructose 6 phosphate. Okay, so this was the whole steps we remember from the video 2. So let's see the options here. Fructose 2 6 bisphosphate production is activated by glucogen. No, fructose 2 6 bisphosphate production is activated by fructose kinase 2 and insulin actually. So this option is wrong. Glucogen inhibits the enzyme fructose 2 6 bisphosphatase. No, it actually does not inhibit its production. Okay, so this option is wrong too. Insulin activates the enzyme phosphofructokinase 2. Yes, I told you that insulin activates this enzyme, and in return it produces fructose 2 6 bisphosphate, which also activates the phosphofructokinase 1 activity. So this option is true. Option number 3 is right. Increased insulin increases the glycolytic production of fructose 6 phosphate. No, this is not true. Okay, so increased insulin does not actually increases the production of fructose 6 phosphate. Okay, so let's see the next question. Which cell in the human body only uses glycolysis for ATP production? So the first option is neuron. So actually here what they're asking is which cell in the human body only uses glycolysis for ATP production. Okay, so no ETC, electron transport chain, and nothing else, only glycolysis. So the answer is erythrocytes. Why? Because as we all know that erythrocytes, the blood cells, they do not have mitochondria. So as they do not have mitochondria, so ETC, or we can say oxidative phosphorylation, to produce additional ATP from pyruvate, they cannot use it. So they cannot use oxidative phosphorylation, okay, because they do not have mitochondria. So they only use glycolysis for the ATP production. Okay, so this was easy. Now, the third question is which statement about glycolysis is correct? So, this is very simple. For example, let's see the options, how it is simple. 3 molecules of NADH2 and 1 molecule of FADH2 are produced? No. During the whole process of glucose to oxidation to pyruvate, we never saw any DH2 and FADH2 production, right? So this option is wrong. The two net molecules of ATP are produced through substrate level phosphorylation. This is correct. Like what we know is during the glucose oxidation to pyruvate, we all know that the four molecules of ATP are produced, right? But We also use 2 ATP molecule in the during the first steps, है ना? So the whole net production is 2 ATPs. So 2 net molecules of ATP are produced through substrate level production. This option is true. So glycolysis cannot proceed under anaerobic condition. This is also wrong. Glycolysis is an anaerobic process and it only takes place in cytoplasm. So it cannot proceed under anaerobic condition is wrong. It is an anaerobic process. Next option is resulting pyruvate molecules are always directly incorporated into the Kreb cycle. No, it is not always necessary. So this is also wrong. So let's see the next question. Which type of enzyme is responsible for initiating the process of glycolysis? We all know that during the whole process, the first enzyme which we see is hexokinase. Okay, so kinase enzymes. The first step is phosphorylation of the glucose, which is accompanied by hexokinase in most of the cells. In most of the cells, hexokinase is responsible, but glucokinase, you will see that glucokinase is also responsible for the glycolysis process, but only occurring in the liver cell or pancreas specifically. Okay, so hexokinase, glucokinase. So this option is true here. So kinase is option which is correct here. Let's see the next question. For eukaryotes, the total yield of ATP from NADH is not always maximized when the NADH is manufactured by which of the following? So, as I told you that sometimes the glycolysis, the ATP produced by glycolysis not always goes to like oxidative phosphorylation, and in return they also produce more ATPs. So it is not always necessary. Electrons sent in the mitochondria to continue the etc. Okay, sometimes the NADH produced, it skips over the NADH dehydrogenase. We all know now that etc, that there are this four complex present. First is NADH dehydrogenases complex. Okay, so not, this is not always true that the glycolysis ATP will go back to the Kreb cycle and there it will produce the NADH and FADH, but that NADH will goes to etc, electron transport chain, and will produce more hydrogen electrons and more energy. So this is not always true. Sometimes the NADH skips the NADH dehydrogenases and goes directly to coenzyme Q. Okay, so it is not always maximized when it uses the glycolysis process. So these are the questions I have prepared from the glycolysis part 1 and part 2. Thank you so much.